Today, we're very excited to have Kevin Bode, PhD. He's a postdoc at HMS and the Joslin in Boston, presenting beta cell engineering to resist and counteract autoimmunity. His bio, a short bio, obtained his PhD in cancer immunology at Ruprecht-Karls University in Heidelberg in collaboration with German Cancer Research Center, DKFZ, in Germany, following postdoctoral training in cell transplantation and autoimmunity at Harvard Medical School and Joslin Diabetes Center. His current projects are related to uncover novel immune regulatory checkpoints in type 1 diabetes, aiming to develop and improve beta cell replacement therapies. His recent paper, a very interesting paper titled Beta Cells Deficient for Renalase, Counteract Autoimmunity by Shaping Natural Killer Cell Activity. I'll drop that URL link in the chat for people's reference. And just a quick little abstract from the paper, because it is, and I'm sure he's going to be going through it, but basically, they previously showed by inactivation of the renalase gene that they could enhance immune resistance of beta cells, and now they demonstrated that renalase loss of function in beta cells shape autoimmunity by mediating natural killer cell phenotype, which is obviously important for the induction of tolerogenic antigen presenting cells. And renalase deficient beta cells mediate cell-cell contact independent induction of hallmark anti-inflammatory cytokine TGF-beta1 in NK cells. In addition, surface expression of key regulatory NK immune checkpoints, CD47 and CCAM1, are markedly elevated on beta cells deficient for renalase gene. And enhanced glucose metabolism in renalase mutant beta cells is responsible for upregulation of the CD4 surface expression. So they really kind of, from this original GWAS screen, identified, in 2009 by Steve Rich and company, they saw their single nucleotide polymorphism in the renalase gene were associated with type 1 diabetes. And then Kevin Bode and his group went further, identified renalase as one of those genes, and sort of dove into the mechanism. So we're really excited to hear from him today. And thank you very much for joining us, Kevin.
Yeah, thank you so much for this great introduction. So I'm be honored to present my major postdoctoral work today to you, about 3.5 years of postdoc at the G Lab in Joslin Diabetes Center. And as you can see, I'm dealing with beta cell engineering and autoimmunity. And first of all, I would to give you a little back.
You froze a little there. Sorry, missed
okay.
that.
Okay, hope it is stable now.
Okay, great.
So it has great potential to make at least the patients insulin independent. But there are big problems like autoimmunity if you use allogenic iPSCs, and of course recurrent autoimmunity because beta cells, they still have beta cell antigens. And for type 1 patients, of course, the memory T cells, they still can recognize these antigens and kill them. So there are ongoing approaches. I think all of you are familiar with the Vertex trial, very promising. Here they use allogenic stem cell-derived beta cells, meaning that they have to deal with alloimmunity and autoimmunity. And of course, they also need to use lots of immunosuppressants to make the cells surviving. So far, I think the results are quite exciting because there are at least 2 patients, they were insulin independent for more than 1 year. I think that shows that in general it's working. But the immunosuppressants, they're a big problem because, I mean, you can think of if you suppress the immune system, you get problems with cancer, infections, and so on. And that's really a big problem. And therefore, I think we and many other groups are diving into genetic engineering to somehow improve the beta cell to survive autoimmunity or even to counteract immune responses. And example approaches are, for example, the induction of immunosuppressive ligands on the surface like HLA-E and CD47. But many groups also using HLA knockout cells to prevent CD8 and CD4 T cell killing directly. But our group, we were more interested in the unbiased approach to find new targets that can be mutated in beta cells and then lead to protection. Here is the screen Monica also introduced already. that we find the renalase gene here. So what we are using is the NOT-SCID NOT transfer model and the beta cell line NIT1, which is a syngenic cell line to NOT. So no autoimmunity, just autoimmunity. We transduced the NIT1 cells with the Gecko library, which targets more than 20,000 genes. And we make sure that only one mutation is occurring, or one guide RNA is transferred to one single NIT1 cell. Then we injected the cells subcutaneously into NOT-SCID, transferred or not, no transfer of diabetogenic NOT splenocytes. And as you can nicely see here in this screen, we have a few hits really strikingly popping up that are enriched, as you can see here, meaning that if the cell got, for example, a guide RNA for renalase to knock that gene out, it has a survival benefit over other genes. And because renalase is known to be a GWAS candidate gene, we focused on this gene for further studies. And just for you to show that here on the left, I don't know if you can see my mouse. So here you can see in the situation of diabetogenic splenocytes, we injected control, so wild type or renalase cells in the same mouse, and then injected the splenocytes. And as you can see here on the left, the wild type is disappearing, but the renalase deficient beta cells, they still survive. And the one major finding of our first paper was that these cells seem to be more stress resistant than the wild type cell, meaning that if you put H2O2 or ER stress on the cells, they tend to survive better than the wild type. And this was also true for stem cell derived beta cells, as you can see here in this approach, where we treated the cells with tapsigarine to induce ER stress, induced apoptosis. And the wild type, you can see there is an increase, but not for the renalase knockout beta-like cells. Okay, so that's nice to know. So we know the cells are more stress resistant. They're surviving autoimmunity better than the wild type. But what is happening with the immune cells in the graft? This was my first aim to look at. And this is the basically approach I'm using for all in vivo experiments. It's similar to the experiment I showed you before. So we injected wild type and renalase cells in the same NOT-SCID mice. We injected NOT splenocytes from diabetogenic mice. And then after different time point, we collected the graft and different organs to look at the immune cell infiltration, immune cell activation by flow cytometry or single cell RNA sequencing. And mostly I'm focusing on day 10 or day 17, because here you can nicely see the graft weight differences between wild type and renalase obtained from the same mouse. And therefore, we were focused on that. I'm showing you mostly data about this time point. And here you can see single-cell RNA-seq gene expression profile data of all isolated immune cells. So we sorted CD45 immune cells and performed 3' single-cell RNA-seq. And as you can see here, on the right side, here you have genes that are upregulated in renalase mutant cells. And one promising factor, of course, is TGF-beta, a very well-known anti-inflammatory cytokine, important for Treg induction or in general to counteract T-cell activation. So this is elevated in immune cells, infiltrating into renalase mutant grafts. And here in black, you can see what pathways are upregulated in the wild type situation. For example, MYC target genes are upregulated, mTOR signaling and oxidative phosphorylation all goes to the direction of immune cell activation. But in the renalase mutant graft immune cells, you can see it's elevated TGF-beta signaling, for example, apoptosis and hypoxia. To go further, we looked at the immune cell profile between wild type and renalase mutant graft, again, obtained from the same mouse. They got the same immune cells, but we got a different cell profile. in terms of CD4 T cell infiltration. So we have more CD4 T cells in the Renalase mutant graft, and also strikingly about half of NK cell proportion compared to the wild type situation. Then the monocytes, macrophages, at least from the general infiltration, are the same also for the DCs. So there's no difference in dendritic cell infiltration overall. And a minor increase in granulocytes and gene expression profile analysis showed that these are myeloid-derived suppressor cells. These are cells known to suppress the immune system in the close environment. And what also was strikingly, we first of all, we got the same data by using flow cytometry. So we saw more CD4 T cell infiltration, less NK cell infiltration. But what we also saw here is that although we have the same amount of APCs, so antigen presenting cells like macrophages and DCs infiltrating into the tumor, into the graft, they show a different phenotype in terms of MHC class II upregulation. So they have reduced MHC class II expression on the surface, and also the levels of MHC class II molecules on the surface are reduced in Renalase mutant grafts. But on the other hand, the well-known immune checkpoint, PD-L1, these checkpoint is increased on the APCs in Renalase mutant graft. So this was not seen by RNA expression, meaning that it's not a regulation of RNA, it's more the availability on the cell surface is changed. So they somehow have more transfer of these molecules to the cell surface. I mean, I think that's important not only to trust your single cell RNA data, protein is also very important and where it's located. So in summary, you can see the Renalase mutant beta cells, they are not only stress resistant, they can shape the immune cell profile. For more CD4 T cells, they're expressing more TGF-beta as well. We saw minor effects on CD8 T cells like PD-1 upregulation, but not much else. We see less NK cell and less activated NK cell and more tolerogenic antigen presenting cells in these grafts. Okay, so because we saw that PD-L1 is upregulated on the APCs, we sought to investigate if this upregulation or PD-L1 in general in this model is important for graft survival. Therefore, we injected in the same or similar in vivo experiment, we injected anti-PD-L1 to block the PD-1-PD-L1 interaction for several times. And then, as you can see on the upper right, the control condition where we just injected control IgG antibodies, you still see the graft rate differences. But for the PD-L1 condition, as expected, the graft is much, much smaller, but you don't see differences in the graft rate anymore, meaning that PD-L1 here plays an important role for the Renalase mutant cells to survive. So we wanted to figure out if it's only the APC or is the PD-L1 also maybe playing a role on the beta cell itself. Therefore, we stained the NIT1 cells with CFSE to follow them and to see the surface expression of PD-L1 on the left downside here. Then you can see directly that PD-L1 is mostly present on CD45 positive cells, so on immune cells, and only a minor CD45 negative cells are positive for PD-L1. And this is also true if you gate on the CFSE positive NIT1 cells. You can see that they barely express any PD-L1 on the surface, even in the inflamed environment. So we knew that the NIT1 cell in general, they do not express PD-L1, but during inflammation, like interferon gamma exposure, beta cells are known to upregulate PD-L1, but not in this case. So the NIT1 cell is not really promising expressor of PD-L1, and there's no difference between wild type and Renalase. But we knew that PD-L1 plays an important role here in the system. Therefore, we thought, okay, maybe we can improve the whole situation, the whole Reynolds mutant graft by putting more PD-L1 into the cells. So we generated wild type and Reynolds mutant NIT1 cells, overexpressing a lot PD-L1 on the surface, and performed in vivo experiments by injecting wild type or wild type plus PD-L1 or Reynolds and Reynolds plus PD-L1. As you can see here, on the wild type, they benefit a little bit from the PD-L1 overexpression, but not as much. So it was surprising for us to see. We expected to see a much better outcome. But for the Reynolds situation, it was even the other way around, although it is not significant. So there is at least no benefit if you put PD-L1 on the beta cells, at least in this NIT1 cell line experiment. So we conclude that PD-L1 expression on APCs rather than on beta cells is crucial for graft protection. And PD-L1 overexpression does not improve the graft survival of Reynolds mutant cells. So the first summary of my talk is we got different immune cell profile and different activation in the wild type of Reynolds mutant graft. For example, we have less MDSCs, myeloid-derived suppressor cells, less activated NK cells, fewer mature MHC class II positive APCs, but more PD-L1 tolerogenic APCs, and more CD4 T cells. Maybe I can tell you, I don't have the time to show you the data, but the CD4 T cells that are enriched, is this a specific CD4 T cell cluster? It is not FOXP3 positive, but the gene expression signature is tending to be similar to a FOXP3 positive Treg in the colon. So maybe they adopt a more tolerogenic Treg-like phenotype, but as I said, not FOXP3 positive. Okay, so after that, we wanted to find out, okay, there is much changing in the graph, but what cell type is really important to induce all these changes? And we focused on natural killer cells because, first of all, they are just they are strongly reduced. We just have half of the cell population in the Reynolds mutant graft. And doing our time course experiments, we saw that NK cell infiltration is reduced very early on, even before you see graft rate differences. So it is an early event, and therefore we think, okay, it's maybe important. And NK cells in the Reynolds mutant graft, they also show elevated expression of TGF-beta in our single-cell RNA-seq data. And the NK cells are more active when they're infiltrated into the wild type. And it is known that NK cells can interact with APCs to regulate the phenotype. And we see that the APC phenotype is dramatically changed here. So therefore, I performed, again, a similar in vivo experiment, but before, in one group, before injecting the autoreactive splenocytes, I depleted NKp46 expressing cells. And this is a well-known NK cell marker. So all cells are present, but no NK cells. Because not SCID mice, they also have some functional NK cells. I wanted to make sure that there are no NK cells around. Therefore, I also use NSG mice that do not have any functional T, B, and NK cells. And as you can see here, if NK cells are depleted, even in the not SCID background, you don't see any differences in graft rate anymore. So that was quite clear. And what is also interesting is that the CD4 T cell infiltration is not dependent on NK cells in the graft, because it doesn't matter. On the left side, we have NK cells. On the right side, we don't have NK cells. You still see elevated CD4 T cell infiltration. But you don't see the reduced NK cell infiltration, of course, because there are almost no NK cells present, and no changes in MHC class II high expressing APCs, as seen before, indicating that NK cells are important to induce the MHC class II in the wild type situation, but it is prevented in the Reynolds mutant situation. And furthermore, what is really important, because PD-L1 was important in our experiments for randomized graft survival, you can see here in the FACS plots that PD-L1 is upregulated on MHC class 2 high positive APCs in the randomized mutant graft, but if NK cells are not there, this effect is totally gone. So it's completely dependent on NK cells. So then we asked, okay, NK cells are important. That was clear, but how is this happening? Is the NK cell communicating directly with the beta cell, or is another cell important to communicate with the NK cell? So therefore, we performed in vitro co-culture experiments with NK cells and NIT1 cells to see are there any direct effects from the wild type NIT and randomized mutant cells on the NK cells. And because we also seen that the randomized mutant cells, they express more cell adhesion molecules than the wild type counterpart, indicating that there may be changes in cell interaction. First of all, we, as I said, we co-cultured NK cells enriched from NOD spleen together with wild type randomized mutant cells for 3 days under IL-2 stimulatory condition. As you can see here in the wild type, the NK cells, they start to express CD107A, which is a degranulation marker and a cellular surface marker for cytotoxicity. And this was strikingly downregulated or even not induced if the randomized mutant cells were co-cultured. So that was quite strikingly. Then we also performed proliferation assays by labeling the NK cells with the tagged violet dye. And as more peaks you see on the left, as more you see NK cell proliferation in the end. And you can see that the proliferation together with randomized mutant cells is drastically reduced. And we also performed similar experiments in transfer to see if this is cell-cell contact dependent. So any one of you don't know what a transfer is, it is kind of a membrane separating NK cells and NIT1 beta cells so that they can communicate with the media or with some soluble factors, but not with cell-cell direct cell-cell interaction. So this was strongly reduced. But on the other hand, for example, the TGF-beta expression, which is elevated on NK cells by co-culture with randomized mutant graft, shows that the randomized mutant cells have a direct effect on NK cells to upregulate TGF-beta, but this effect is not dependent on cell-cell contact. As you can see here, the differences are still there, even though you separate the cells by a transfer. So to sum this up, we can see direct effects of NIT1 randomized mutant cells on NK cell activation, which is cell-cell contact dependent or independent. So they have different mechanisms to regulate NK cell activations. So, but how is that happening? How can the NIT1 cells do that? So we performed RNA sequencing of randomized cells against the wild type. And as you can see here, we only focus on immune cell-related genes. You can see that, for example, CD44 and ITGA4, which are cell adhesion markers, these are upregulated in randomized mutant cells. But you can also see that very well-known master inhibitor of NK cell activation, CCAM1, is upregulated on randomized mutant cells. And furthermore, not that strikingly, but significantly upregulated is CD47, which has been shown recently to be effective in suppressing autoimmunity or autoimmunity against hyperimmune primary pancreatic islets. So, and this is also known to be a main checkpoint to suppress NK cell activation. So it is really nice to see that this is all working in NIT1 mouse cells, but of course, in the end, we are interested in human cells, and therefore we wanted to see if this all translates to the human system. Therefore, we used iPSCs, wild type and randomized mutant, and differentiated them for 35 days to stage 6 2D beta-like cells, and perform co-culture experiments with HLA mismatched PBMCs in a stimulatory condition. As you can see here, the differentiation was similar between wild type and granulase, so there's no difference in beta cell generation. And on the bottom left, you can see FACS plots showing the CD47 expression on the y-axis and the CD66A expression on the x-axis, which means CCAM1 from different control cells and from wild type and granulase mutant single cells, which are C-peptide positive and NKX6.1 high positive. And you can nicely see that the granulase mutant cells, similar or the same way as NIT1 cells, they start to overexpress or have elevated surface expression of CD47 and CCAM1 together. As again, very important inhibitors for NK cell activation that makes sense. Here you can see the quantification, and interestingly as well is that staining the NK cells in the PBMCs for TGF-beta or for membrane-bound TGF-beta, you can also nicely see increase in TGF-beta expression on these NK cells, recapitulating the same results we saw before with NIT1 cells. So that was strikingly. And although the killing is not that efficient in this assay, because we co-cultured only for 3 days, we still see there is a difference in beta cell survival of wild type and granulase mutant cells we also saw before. Okay, so that sounds promising. Obviously it is translating to the human system, but what is the mechanism? How is CD47 or CCAM1 upregulated in the beta cell? And by performing RNA-seq and seahorse experiments, we already knew that there is a strong hint that glucose metabolism or glycolysis is upregulated in the granulase mutant cells, shown by enrichment analysis for pathway and for related pathways, and by the acidification rate in the seahorse experiment. So therefore, I make use of a glucose metabolism inhibitor called 2DG, which can be taken up by the cell like glucose, and can also be phosphorylate the first step of the glycolysis, but then because of the H atom here, which normally is a OH, it is stocked, so it cannot be further consumed by glycolysis, meaning that glycolysis is inhibited by using this drug. And also other pathways like pentose phosphate pathway could also be altered here, but it is known as the main glycolysis inhibitor. And here I use different concentrations of 2DG from 1 to 10 millimolar. And you can see here that by 3 days co-culturing the cells with the drug, you can see that the surface expression of CD47 is decreased. And this is also true for CCAM1. So it has been shown that PDL1 or also CD47 are similar regulated in terms of expression. So it was not a surprise that CD47 can be reduced by inhibition of glycolysis. But for CCAM1, that's completely new. So I don't find any literature about that CCAM1 expression is regulated by glycolysis. But at least it is kind of a hint by using 2DG that glycolysis plays a role here. And first and last of all, these are unpublished data and just one experiment, so it has to be repeated, but I mean, the results are very strikingly, therefore I want to show you the results here. So we used wild type and granulase mutant NIT1 cells again, and treated them with a low dose of 2DG to make sure they have a similar glycolysis rate. So that they have both suppressed I'm sorry. So I just treated the renalase cells with 2DG or not to compare with the wild type cells to decrease the glycolysis activity and make it more wild type like for 2 days, sorry. And then I reseeded the cells to make sure the 2DG is gone and it's not influencing the NK cells. Then I put on the NK cells from autoreactive splenocytes, co-cultured for 3 days with IL-2 and performed flow cytometry analysis. And as you can see here in the pinkish color, these are the wild type like renalase mutant cells treated with 2DG. And they tend to increase CD69, which is an activation marker again, similar as the wild type. And that's also true for CD107A. So the suppression is reduced, meaning that they are kind of wild type level. And this is also true for TGF-beta expression, meaning that reduced glycolysis reduces the TGF-beta expression of NK cells. So yeah, this was really striking that maybe the change in metabolism in the beta cells can influence the cell surface expression of several checkpoint inhibitors on the surface, then altering the NK cell activation and overall the autoimmune response, as you can see here in the summary. What are our future directions? So we are also interested in studying the CD4 T-cell mediated beta cell killing because it is widely ignored by many people, but it is well known since many, many years that CD4 T-cells alone can kill beta cells. You don't need CD8 T-cells. I mean, they can do it maybe faster, but CD4 T-cells can do it as well. And they don't need MHC class 2 for that on the beta cells. And first of all, we wanted to check our NIT1 model system. How is that killed? CD4, CD8 mediated. And strikingly, if you deplete CD4 T cells, the killing is almost gone. So there's a little bit killing of the CD8 T cells. It is significant, but by comparing with CD8 depletion, where we only have CD4 T cells, the killing is massively increased, indicating that our cell model is suitable for investigate CD4 T-cell mediated killing. And I mean, it was not that surprising because we know the NIT1 cells, they do express a low level of MHC class 1. So they are not that accessible for CD8 mediated killing, probably. So that was not a surprise, maybe here. But CD4 T cells can do the job nicely here in this model. As you can see here, the depletion was successful. No CD4 T cells, when CD4 T cells were depleted with an antibody in vivo, and CD8 T cells were also nicely depleted in the graft. Then furthermore, it is also known for several years that CD4 T cells alone can transfer disease into non-SCID mice. Here in black, you can see polyclonal CD4 and CD8 T cells. They can induce diabetes in 100% of the mice in 30 days, so they can do it faster. That's clear. But CD4 T cells alone, where CD8 T cells are depleted, I make sure they are depleted. So I stain them, the pancreas, and they are not CD8 T cells anymore. They are killed after some time. It takes longer, but about 40% to 50% of the mice develop diabetes. And this is accelerated if you inject anti-PD-1 into the mice. But more strikingly is the transgenic model of the BDC 2.5 CD4 T cells. If you inject these cells into the not-SCID mice, they are even more aggressive than the polyclonal CD4 and CD8 T cells. So starting after 10 days, the mice become diabetic. It's very quickly and very aggressive. And you always only have CD4 T cells here. And therefore, we would like to make use of this system, so BDC 2.5 immune cells and the NIT1 beta cells which we want to again make a CRISPR-Cas9 screen using a different library covering more genes. And but not only restricted to CRISPR-Cas9, we also would like to do an open breeding frame induction assay by seeing what genes in beta cells can be elevated to protect against autoimmunity mediated by CD4 T cell killing. And as you can see here, you have to check, of course, it's important that the NIT1 cells can be killed by BDC 2.5 CD4 T cells as well. And you can see it here. Okay, then finally, I would like to thank all of you for attention. And of course, all collaborators and funding resources like the GDF, the Center Grant, and the whole Kissler lab, which I started my postdoc in, but now Stefan Kissler is at Vertex, leading the immunology there. And I'm now with Yi Lab, and he's a very expert in CRISPR screens and genomics. And I learned really a lot in both of these labs. So yeah, thank you very much.
Thank you, Kevin. That was really, really fascinating. And you've made a lot of progress in this system. Opening up for questions from anyone else. If you'd like to have ask a question, please raise your hand or unmute yourself or drop something in the chat. Okay, anybody? No? I had a couple questions for you.
Mm-hmm.
So just to head back towards renalase. Renalase, right, is a flavonoidenine dinucleide domain containing protein, right? It functions sometimes as a cytokine, sometimes as an anomerase. It's it's emerging like it's a novel regulator of cardiometabolic diseases. It's really expressed or secreted by the kidneys, and it's been reported to have this like hypotensive effect and controls blood pressure through regulating sympathetic tone. What could you say about the role of renalase removal, I guess, in regulation of the sympathetic tone as it pertains to the sympathetic innervation of islets in their vasculature? This would be like, in vivo, not in a stem cell environment.
Okay, so yeah, first of all, the function of the renalase gene, I think it's still kind of mysterious. There are some findings showing the potential substrate of renalase, but this has been disproved later, or is just proved in silico and not in vivo or in cellular systems. So it's really not clear, first of all, what is the substrate of renalase and what is the real function in the cell. It's probably like the conversion of NADPH to a functional NADPH. But that's still not clear. So I'm really interested. What is the real function of renalase?
Yeah,
my former colleague Tara McDonald is trying to solve that issue. She has now her own lab in Toronto to figure that out, but it's still unclear. But she performed a lot of experiments in islets, in stem cell beta-like cells, and in NIT cells. And all we can always see is that the glucose metabolism, for example, and amino acid availability is changing in the knockout of the renalase mutant cells. So they definitely have a role in metabolism, especially glucose metabolism. The role, renalase is normally produced by the kidney and not really, in a physiological way, by the kidney to perform the hypotensional function, right? I'm not sure what about this function in beta cells?
Yeah, they have beta cells have the alpha-2 adrenergic receptors, right? So it's kind of interesting.
Yeah, that's true. I don't know. You think it's maybe counteracting the vasculature formation?
I don't know. Joanna Almaca might have something to say if she was on the call. She's all in with the pericytes and the role of the vasculature. But that's for another day, I guess. But the other it does, sort of in reading up on renalase, it looks like it's secreted as a proenzyme and it can be activated by catecholamines, right? So there's some catecholamines in the natural environment of the islets. And that it looks like it doesn't only degrade the catecholamines, but it also metabolizes other substrates. So it's very interesting. Todd Briscoe has a question. Do the molecular inhibitors of renalase induce the changes on CCAM1?
Oh, that's a good point. So yeah, we have a very broad and not very specific renalase inhibitor. So this also inhibiting MRO, not only renalase. And I tried it once or twice to see if the inhibition of renalase by this inhibitor called pargolin is inducing these kind of effects. And so far, I think it is recapitulating the slide is recapitulating the CD47 downregulation, upregulation, but not to a strong extent. So just a minor increase, just a tendency. So it's not really recapitulating the renalase knockout so far, but we think it's maybe because it's too unspecific. So we are also searching for a new drug that is more specific to inhibit renalase. So there's still a question mark, but we are on it. So that would be really cool.
Okay. Sorry about that. Here, I have another question. What can you say about how renalase has been shown to be protective in the pancreatic acinar cells through the plasma membrane calcium ATPase? And in doing so, it reduces pancreatitis. So what do you think of, like, through this removal of renalase from the beta cells, could the beta cells also have an effect on their plasma membrane calcium ATPase?
I mean, this you would also see as a consequence in insulin secretion, right? If there's a problem with calcium influx, and so far we don't see differences in insulin expression.
Yeah.
So
you think
I think it's probably not a problem, but I think we have to do more experiments to really rule that out. So
yeah,
that's
it's
for
interesting.
sure.
It's just kind of curious. It's it's a little, that renalase is a little bit of a black box still, but it does seem to be working in your system. So that's exciting.
Exactly.
I just have two more questions. One is, does this have a potential for earlier therapeutics in T1D stage 1? Like, could you target beta cells with renalase blockers, or would you have some off-target effects to be concerned with?
So we performed experiments in not-skid mice. We gave them pargolin in the drinking water, and it was sufficient to reduce killing in the NIT1 model. So that in general, it's working. We also tried similar experiments, but as you can see, it's more time consuming by treating the NOD mice very early on with pargolin, but you have to change it every 2 days. It is not that stable. So we did that, and there was just a tendency. It was not significantly protected. I mean, probably if we have a more specific and better drug to target the renalase with a lower concentration, then I think we can repeat this kind of experiments to see if this is really can be used as a prevention drug. But so far, it is not clear.
Yeah, it's kind of interesting because, verapamil now is also a blood pressure medication, sort of in the same, under this, not the same umbrella, but a similar umbrella, right? Because this also affects blood pressure. And then you've got some protective effects. So that's really curious. My last question is, what do you think about a genetic screen in newborns that could ID the SNPs in the renalase and then kind of like bin those people into an endotype at risk for type 1 diabetes? I mean, there's definitely been seen that there's some renalase genetic, SNP differences in those that are at risk for type 1.
Exactly. I think I'm not sure if this has been shown completely, but as I know that these mutations are kind of gain-off mutations. So that you have more active renalase, I think even more renalase expression. And this is then indicated to have higher risk for T1D development. Exactly.
Yeah, I think that, some of that, the 2009 paper by Steve Rich and company, they did show that signal nucleotide polymorphism in the renalase genes are associated with type 1 diabetes. So maybe that's a way to identify those that would benefit for this type of approach. So,
That's true.
yeah.
Yeah, very good idea.
All right then. Well, if there's any more questions, anybody else? Last call. Thank you again, Kevin. This is really exciting. And we continue to watch as you progress with this work. And it looks like you're kind of in partnership or loose partnership with Vertex and that group. So that's exciting too. And we'll be in touch. Thanks again for joining us.
Thank you so much.
All right. Have a great rest of your day. Bye-bye.
Bye-bye.